Hello, good afternoon.
Hello.
Hello, good afternoon, Todd. How are you?
Good morning.
Morning. It's more than 12, and for me, it's afternoon as well. Okay, I'm going to Isabel is going to join as well in a few minutes. We already have somebody on the waiting room. I am figuring out how to give co-host rights. Okay, let's see. Make co-host. Okay, so you are a co-host already, and now I will make everybody else co-host. Usually it's only for sharing screens, so I'm not sure if you need, I don't think you need it, but the Q&A is right there. So I'm just going to admin Inge Mulder. Hello, no, still connecting. Hello. Good afternoon. Hello. I can't hear you. No.
Hello.
Yes, perfect.
Yes. I had to change some settings.
No problem. So we have you finally here. Good afternoon.
Yes.
How are you, Dr. Mulder?
Good, good. Thank you.
Okay, I have given you already the rights to be a co-host.
Okay. Do you
Which
want to
gives you,
test this?
yes, it's really as per your comfortability if you want to test your share screen.
Where's the button? Oh, here, share. You should see it now, right?
Yes, we do.
Okay, perfect.
Perfect. We wait for the others to join. Meanwhile,
So how often do you do these webinars? You organize that?
for Microplastics Initiative, we have 4 webinars in line for this year. So we've already done one webinar in March, end of February, March. And now this is the second one in line. We aim to do another one somewhere around September. It's still under discussion, but we for sure will be doing an in-person workshop here at LC office in Brussels on analytics of microplastics.
Oh, yeah.
And then followed by December, the last webinar of the year. So it's more on the community of practice and spreading knowledge.
Yeah, nice.
Is it your first time with such webinars? Have you been
Yeah.
affiliated with LC before?
No.
No? Okay.
No, I was invited to do this via a colleague of mine.
Marja.
Maria.
Yes. Yeah.
Yeah, she was
Yes.
like, "Oh, if it's BBB, then I should ask you and not do it
Thank you.
myself. Yeah.
Thank you very much. Hello, good afternoon, Philip.
Hello, do you hear me?
Yes, we do. Loud and
Oh,
clear.
thank you. I wasn't sure if it works.
It works. Good afternoon. How are you, Philip?
Yeah, I'm very good, thanks. How about
Great.
you?
Great, very well as well. I have given you the co-host rights as well. You can share your screen, and if you would like to test the screen sharing on
All
Zoom
right.
as per
try.
your comfortability.
I need to find out how to do it.
Uh,
there is the button. Okay.
the
Yeah,
green one.
okay. Yeah. Oh. Okay. So you see me sharing now?
Yes, the entire screen actually, not the presentation.
Yeah, I will try with the presentation now. Okay. All right. How about now?
Yes, we do.
Okay.
Yeah. Hello.
Good afternoon, everybody.
Hello.
Good afternoon. Just gonna close the door.
Good afternoon, everybody. Gus, I have made you a co-host as well. I'm not sure who's going to share the screen from you or Dr. Been.
That will be me.
Okay, great. Then I think you can test the screen however you feel like at the moment. And see if it works for you.
Yes. Wait, I'll just.
Good afternoon, Marianna.
Hello, everyone. Can you hear me?
Yes, loud and clear,
Yes.
Marianna.
Okay.
Yeah.
Great.
This should be clear, right?
Yes, it is. Thank you, Gus. Marianna, I have also made you the co-host to share your screen. If you would
Yeah.
like to check
try.
it out. Yes,
Yes.
we still have time.
Let me see. Can you see my screen?
Yes, we do.
Okay, great.
Not yet on presentation mode though.
Yes.
Sorry.
It was not on presentation mode though, but maybe you haven't clicked it.
Okay, let me try again. Yeah, it was not on presentation mode. Does it work now?
Yes, it does. Thank you.
Okay, thank you.
Fuji, do you want me to share the screen or do you want?
I think a major part is from your behalf, so please go ahead.
Okay, wonderful. Then I'll click forward for you when you're ready.
No problem. We wait for Dr. Federic and then we can admit the others.
I send them a message just now.
No problem. Still in time.
I was just going to inquire, I don't seem to see a Q&A.
No, we don't have that function indeed. We only have the chat function. Yeah.
Questions.
So there will be no filtering of the questions in advance, but of course we can pick questions. Who is moderating the Q&A? Is that you, Todd, or is that you? Yeah.
It's Todd.
Yeah.
Yes.
So Ruchi will give the practical guidelines on saying that no need to take the recording will be made available, the slides will be made available, and we have a Q&A at the end of the talk, and stimulates people to put in the chat the questions while the presenters are ongoing. So then, Todd, you can pick which questions that you want. So if there's any question that you want to skip, you can say that due to the lack of time, we will address your question by email, for example. That could be a way of doing it. Yeah.
Okay. So I just handle the question aspect at the end. Perfect.
Yes. Yeah. But we can also maybe during the presentations, Ruchi, we can also put in the chat. Feel free to put your question, drop your question here while the presenter's going. So, but to the presenters, please ignore the chat. You don't need to look at it. Just keep on going. Yeah.
And will you do the questions all questions at the end or after each presentation?
Just in the end.
Okay.
Right in the end, we have a 10-minute session.
It's not a lot, to be honest.
Yes,
No.
we're running very tight.
Yeah.
You might have to be very strict on questions, I guess. Have to prioritize in case there's a lot. Okay, perfect.
So if you want to go shorter on your presentation, you can. You cannot go over time, though. That wouldn't be recommended. Indeed, we need to leave some time for Q&A. Yeah. We have quite okay registrations, around 30 registrations. So good audience. Not sure who. That's maybe the fireflies emoji. Maybe that's
Okay.
if you want, I'll go and check.
Or is it a possibility that Dr. Federic tried to enter not Zoom, but here?
Yeah, that could be. Shall I go to the Teams and say it and guide her here?
Yes, please, because then I cannot leave the first presentation. Thank you.
I'll leave and then.
We have a notification on the Teams that somebody tried to join the Teams invite.
Can I forward my my Zoom link?
Yes, it is Dr. Federic. He is one. Yes, you can.
We can maybe put it in the chat.
Yeah, that would also work.
Yeah, indeed. I'll go there. It says she does not have the link. Can I share? Where is it?
There are also 4 people in the waiting room.
It's not admitting, but I would rather wait for Isabel because she has the host presentation. So
Frederic is a he, by the way.
great, perfect.
And of course, my computer does not like 2 systems at the same time, so my Teams didn't work. So it didn't let me in. I did deny her access. And I saw then afterwards that you sent a message as well, Fuji. I'll try to see if I can send
I
you.
have 2 Cas, and I hope it's the other one is Frederic.
Yeah, it's the other one.
Okay.
Maybe Uchi.
Hello, everyone.
Good afternoon. Hello.
Good afternoon.
Yes,
Maybe you can remove the link in your Outlook invitation and say Z Zoom or use a Zoom link.
Isabel, he is here.
Ah, wonderful.
Okay. So we have
Somebody-
problem solved, and now we can let the others enter. If everybody is in line, we go ahead. Is the green signal?
Mhm.
Then great. Let's move on.
Welcome, everybody. Good afternoon. We give it a few minutes before we start. So. Good afternoon for those who joined.
Hello. Hi.
Hello. Hello, Isabella. Welcome.
Nice to see you. Thank you.
Yeah, several familiar faces, so welcome all. I propose that we start. So good afternoon. It's really my pleasure to welcome you to this ILSI Europe webinar, which is entitled Microplastic Quantification in Human Blood and Bioaccumulation in Human Brain. My name is Isabel Hullings and I'm the Executive Director at ILSI. For those who are not yet familiar with ILSI Europe, allow me to give you a brief introduction and also say a little bit more about the Microplastics Initiative, which is the initiative behind this webinar. So ILSI stands for International Life Sciences Institute, which is a global non-profit federation, and you can see here on the screen the different branches across the world. Our mission is to provide science-based guidance to the food sector, and this by providing a neutral platform where we foster collaboration between companies, academics, and the public sector to discuss different stakes, different point of view, and to find a consensus between all the stakeholders based on science. Our vision is that indeed decisions are driven by data and science consensus. We address pre-competitive topics in the area of food safety and quality, but also nutrition and health. This webinar today is linked to the Microplastics Initiative, one of the food safety initiatives. We wish our science to be guiding decisions. That's why we aim and have an operating model that follows the following principles that you see here on the screen. The target of these principles is really to ensure that the science we disseminate and put forward is of the highest qualities, is with every transparency possible, is also independent and inclusive for everybody's opinion. And this webinar will not be any different. For example, every speaker will provide a conflict of interest or declaration of interest behind them. And also we wish to aim for the highest based science there. So that's why we also critically assess science that is put forward in the state of the art. Indeed, our deliverables are peer-reviewed publications, but it can also be black and white documents, as you can see here on the screen. These are more standard operating procedures. And then we also have other deliverables like you can see. A little bit more about the Microplastics Initiative. The participants to these initiatives have one goal, a common goal, which is ultimately to safeguard the human health. And this by advancing as much knowledge as possible about micro and nanoplastics. And the focus is here specifically on food and beverages. So some of the activities they have done is they've initiated indeed this community of practice, which I will say a little bit more about it. But the aim is also that this group will initiate, develop and manage activity proposals and also deliver the science on it. On our website, you can find more deliverables that already are developed by these initiatives, such as other webinars and communication materials as well. All publications are free access on the ILSI website. A bit more about the community of practice. So really it's about providing a neutral platform where professionals, researchers and experts can exchange on scientific knowledge. As I said, the aim is here to drive and find the highest quality standards. So that's why we have to be critical, but constructive to each other. Every research question is welcome, and we discuss how we can address it in the best possible way, which methodology to use, and so on. The community of practice is also about creating synergies and connecting ongoing initiatives, and also possibly to address together gaps that still need to be addressed. The community of practice has a focus on 4 areas that you see on the screen. So it's about analytical methods regarding microplastics, but also exposure for the human, the health impact, and also the biomonitoring. Today, this webinar is really about this first objective around constructively and critically exchanging on scientific knowledge, recent knowledge that has been published. That's why we will zoom in on recent publications regarding the topic of the microplastics. And with this, I'll hold it in the introduction and I'll hand it over to my team member, Ruchi, who will say a bit more about the aim of this webinar specifically.
Thank you, Isabel. Indeed, we will be discussing 2 webinars, 2 publications today. If you could please change the slide. Here we go. So we will be discussing two publications today, one of them being improved multivariate quantification of plastic particles in human blood using non-targeted pyrolysis, GCMS, and bioaccumulation of microplastics in distant human brains. We have the pleasure today from hearing from key experts in the field, beginning with the topic on plastic particles in human blood, new insights from multivariate quantification, led by the authors of the paper, Dr. Federic Been and Dr. Cas Hultes, followed by a session led by experts of Haleon, Dr. Marianna Armogida and Dr. Filip Sucharski, on blood-brain barrier overview and understanding of the methodology used in the publications. We will be wrapping up this webinar with an expert session by Dr. Inge Mulder, speaking on crossing the line, microplastics and their potential to breach the blood-brain barrier. We will be following this up with a Q&A session, moderated respectfully by Dr. Todd Gwynn. For some practical information, we will be recording this webinar, and the slides will be available on ILSI website and the COP Community of Practice. During this webinar, not to interrupt the speakers, we request you to send your questions via the chat, which is open for everybody. Thank you. I give the floor now on to Dr. Been and Dr. Cas, please. Feel free to share your slides.
Us will be giving the presentation on our behalf.
Yes, I hope everyone can hear me
Absolutely, thank you.
and see the slide. So I'd like to start with this declaration of interest slide. Me and Fred are both employed by the Vrije Universiteit Amsterdam, so it's a university in Amsterdam. While Fred is also an employee of KWR, which is a Dutch water research institute. And the research that we will present is funded by Zon en Wee, which is a foundation responsible for funding health-related research in the Netherlands. You can see the details of this research here. It's part of a Momentum, which is a Dutch consortium for micro and nanoplastics research. So with that, I'd like to move on to our presentation. So I will be discussing a little bit the publication that was already mentioned, but I would also like to spend a little bit more time on the background of this kind of research, so that I hope that will
Seems like we've lost Cas.
with these two pictures.
We lost you for a second, Cas, but now you're back.
Okay. So this review from the Bart Koolmans group really summarizes this well. So we have the exposure pathways or the sources of microplastics here on the left, with kind of an indication of what their contribution is. And on the right, we see the typical exposure pathways for humans. So that's inhalation and dietary intake, but also dermal absorption. And this is an overview from last year where microplastics have been found in humans. And this is already outdated now because we've detected it on a lot more organs or places in the human body than just shown here. So how are these MNPs actually quantified in humans, but also other samples? That's basically two main categories of techniques. We have the microscopic and spectroscopic techniques, which mostly rely on particle counting. So you count how many particles are present in a sample. And there's the spectrometric techniques, like pyrolysis GCMS. And there we don't really count the particles, but we weigh them as a matter of fact. And I will explain a little bit more how that works. So we focus on this technique here on the right, which has also been used by the Brain paper. So what is pyrolysis GCMS? In essence, what we do with pyrolysis is that we burn our sample, and in that sample are plastics. And when you burn plastics in this way, they fragment into smaller pieces, smaller molecules, and these we can measure with gas chromatography and mass spectrometry. So we use these pyrolysis products as an indicator of the presence of a plastic. And then to quantify a plastic, we can make a calibration curve. So we spike known amounts of plastic in a sample, and then we measure, for instance, the amount of benzoic acids that we produce during the pyrolysis, and this allows us to put kind of the amount of plastics versus how much benzoic acid we detect. This gives us a calibration curve, and then from new samples like blood samples, we measure them in a similar way. We then detect how much benzoic acid is there, and that tells us indirectly how much plastic was in the sample. We can't just put a blood sample in our machine. We actually have to pre-process this sample. So for the work that I'll be showing, and which is the routine approach in our lab to analyze blood, we take 1 ml of blood and we do an enzymatic digestion to make this blood, to break down the proteins and such in the blood. Then this allows us to also be able to filter the blood, and we do this on a filter with a pore size of 700 nanometers, meaning that we capture all the plastic particles that are higher than this pore size on a filter. But this filter is not completely clean and not just contains plastic particles, but may also still contain some blood residues, which I will talk about a little bit more later. And then we analyze this with our technique. So it's good to realize that pyrolysis GCMS and the analysis of micro nanoplastics in humans is not standardized at all. So there's many different approaches, especially in the digestion phase, but also some groups use filtration, others use centrifugation and then measure a pellet. And it's not surprising that this is not standardized because we're dealing with lots of different matrices. So a blood sample may require a different kind of sample cleanup than a brain or an artery. So here's a quick example of what then is found in different matrices. And what stands out is that the quantities that are being detected vary very much between samples. And that may be just because bioaccumulation is different in these samples. But as I showed, there's also lots of different, there's big differences in methodology. And this is a bit of an unexplored topic in this field. So what's, for instance, not well known currently is what is the effect of a sample matrix on this pyrolysis process. Effectively, we're burning a plastic in the presence of many other molecules that are a residue of these residues of these matrices at a very high temperature, and you may imagine that there's effects from this matrix. So maybe some markers go up in intensities and maybe others go down, which would distort the response that you measured, and thus leading maybe to false negatives or false positives. So that's basically, this is basically our paper in brief. So to investigate this effect, we used spike blood. So we added plastics to blood deliberately so that we could monitor what happens to the plastic signals that we measure because of the presence of blood. Then we used our technique to analyze these samples, and then we looked at these 3 different things, so matrix effects, but we also looked at which features, so which pyrolysis products are really the most reliable to quantify microplastics, and can we use the information of multiple of these products together to come to a more accurate quantification of the plastics. So I will start discussing with we'll start with the matrix effects. So I would like to take polyethylene as a first example. So from already literature research is known that polyethylene breaks down in these molecules called alpha alkenes, which are also long chains of hydrocarbons, but in fact shorter fragments from the polyethylene itself. And what some earlier research also has shown that polyethylene is not the only kind of molecule to make these alpha alkenes, but they can, for instance, also be produced by fatty acids, which on a molecular level look kind of similar to polyethylene. So to attest this effect in the samples that we measured, what we did is we looked at this matrix effect, and that is basically what is the response of the molecular markers from polyethylene, how is it affected by blood? So we measure the response with blood there and with blood not there. And we look how well, so this matrix effect is shown here on the x-axis. And we looked how well is every dot, how well can you use it to explain the quantity of polyethylene. So every dot here is a molecule that we detect with our technique. And you can see here that the dots here at the top are alpha alkenes, so these known markers for polyethylene. They correlate well with the quantity of polyethylene, and the matrix effect is about 100%, meaning that the quantity that we measure of them is not affected by the presence of blood. For instance, here all the way to the right, we see that there is also compounds in the sample that are only abundant in the blood sample and that do not correspond to polyethylene. So these are just the molecules that are maybe still residues of the blood present in the matrix. So this is good news, actually, what we see here, because it means that PE is minimally affected by the presence of blood. So there's no copyrolysis, as we call this, and that the alpha alkenes are actually not markers, and that we don't have to worry about false positives arising from these fatty acids, essentially. We looked at this effect for another polymer as well, for polyethylene terephthalate. So this polymer is known that it's pyrolyzes to these kind of products. And then if you make a similar picture for PET, we see that this looks a lot more complicated. And what that means, what we think it means, is that PET, not in the presence of blood, not only forms these known markers, but also new markers. And that's are the markers that are shown here with nitrogen in their structure, so the N, because this nitrogen has to come from somewhere else than just the polymer, because there's no nitrogen present in PET. So likely this polymer is reacting with the blood or whatever is left of the blood, such as proteins, in this high temperature reaction chamber, essentially. And it means that we have to be careful when we do this quantification of PET, because the markers that we typically rely on might be severely less present. So for instance, we see a line here at 50%. So that means that only 50% of our markers is left, and we might think that only 1 microgram is present, where in fact it should be 2. To determine what is then the best marker for PET, we would look up what's high up here and what is close to 100%. So that may be, for instance, this molecule here, acetophenone. Now, to look a little bit more into this quantification and how we can improve it, we did a follow-up work in our publication using multivariate quantification. So that is where you look at not just one marker. So we don't just rely on, for instance, acetophenone, but we take the information of multiple markers together and translate that to a polymer quantity. So to kind of give you an example, I won't go into too much detail on the chemometrics behind this approach, but we can kind of plot how accurate our quantification is. So basically taking these percentages, the lower the better, because that means a lower error. So this is when we rely on just single markers, but what we can do is use partial least squares to combine all these markers together. And we see that in this case, it doesn't really improve our model so much, but that's because we've now combined the information of all the markers that we detect, but not all of these are necessarily related to polyethylene terephthalate. So what we can do, and that we use 2 different techniques, Boruta and recursive feature elimination. I won't go into too much detail, but these methods basically do is they throw away bad features, as we call them, or signals that do not originate from the sample and only introduce additional errors. And when we do this, we can reduce the model error, so improve the accuracy of our quantification, but also make the model easier to implement because we have to rely on fewer markers that we used for the quantification. So we see that this approach improves the model accuracy, and we've also benchmarked this against another machine learning approach called random forest. And we see also that, yeah, random forest is not as well in quantifying PET, and so we decided to stick to this partial least square model. So yeah, I'm already running, I think, late a little bit on time, so I'd like to summarize the results that we obtained. So here on the right, you see how this quantification in the end improved our quantification of these three polymers, so PE, PET, and PVC. We see a consistent improvement when we rely on this multivariate approach, so using multiple markers. And also our approach of looking at the effect of the matrix effect allows us to kind of say something about how markers are influenced by the presence of blood. And we see for PE no real interferences. For PET, we see that it undergoes secondary reactions, thus you should be careful with quantifying this polymer. And for PVC, where it also was known from literature that there might be interferences, we didn't see this so much, but it was more difficult to quantify due to higher variations in our data. I think what's most important is this take-home message that pyrolysis is a very complex technique where a lot of things can have an influence on the results that you get. And therefore, it's very important to do an extensive method validation before you jump to conclusions. So this means, yeah, work with plenty of negative controls, but also positive controls, as I've shown you here. And really, yeah, go into the details of your method to see what you may be measuring and what may be coming from something else. So with that, I would like to thank my co-authors, and especially Wilco Nijenhuis, who's a PhD student in our group who really led this project and did a lot of work on it. I'll be open to questions, or we do this during the discussion.
Thank you very much, Gus. Indeed, we will take some questions in the end of the session. With pleasure, I would like to invite our next speaker, Dr. Filip and Dr. Marianna from Haleon. Please feel free to share your screen.
Yeah, thank you. Oops. Okay, hello everyone. So this is, let's get started with the declaration of interest. Both Filip and myself are full-time employee and shareholder of Haleon. So today I'm going just to give you a quick overview of the blood-brain barrier, and then Filip is going to share his understanding of the methodology used in the bioaccumulation of microplastic in human brains, Nature recent article. So just to say high level a summary here. So the blood-brain barrier is a complex structure. The primary purpose is to protect the central nervous systems from harmful substances, but also it has a physiological role to maintain homeostasis, to facilitate the transport of molecules and regulates inflammation. It's made of cells and junctions, and it's very tight in the sense that only small hydrophilic compounds, less than 150 DA, or highly hydrophobic compounds, less than 400, 600 DA, can get through the barrier. So in terms of structures, in terms of cell, the first layer is made up by the endothelial cells. They are really responsible for the diffusion of molecules from blood to the brain tissue. Then we have the astrocytes. They are central nervous system cells. They're really the backbone of this barrier. And then in between pericytes, they are mural cells with the purpose of communicating closely with the endothelial cells, as well as they have an impact of the number of tight junctions and the shapes of the astrocytes. Tight junctions are really the gates, for the transition of these essential molecules, and adherent junctions are responsible for the structural integrity and the appropriate assembly of the tight junctions. Both tight junctions and adherent junctions are made of proteins. In terms of physiology, yes, just again to say that the purpose is really to maintain a controlled microenvironment here. So not only to protect the brain, but also to ensure that the molecules and the ions are appropriately, let's say, delivered into the brain. There is a kind of homeostasis in terms of balance of these substances. And this is done via a regulation of ion channels as well as transporters. Mainly active transporters that are responsible for the transport of essential molecules, metabolites, and other nutrients. So here I just wanted to touch base on the drug delivery route. When the central nervous system, the brain, is the target of a pharmacological treatment, this is the options we have today. The first one is passive transcytosis, which uses the natural mechanism. However, we have also said that this is useful only for small lipophilic molecules. So the limitations there. Then we have intranasal administration pathway. This is possible via the olfactory and trigeminal nerve pathways. And this can help delivering proteins, peptides, and other large molecules. Another option is via ligand conjugation. So the drug is attached to a ligand, and the ligand is recognized by a receptor, and therefore this is internalized within the central nervous system. There are also membrane coating strategies. So the coating is made up by encapsulating drugs in carriers like liposomes, nanoparticles, or vesicles. They are going to merge with the cell membranes for delivering these large molecules. And the membrane coating can also allow some, let's say, some strategies in which the autoimmune system is bypassed, like these membranes could be derived from red blood cell or leukocytes. And then we have nanotechnologies, which employs nanoparticles, liposomes, nanogels as carriers to really deliver a different range of agents. And finally, external factors like light and ultrasound can be used to temporarily, break the junctions, the tight junctions, to allow the drugs to get through the barrier. And finally, here I just wanted to show you the sites of these drug delivery systems. As you can see, they are all in the nano range, which is in line with the nanoparticles of polyethylene, which have been noticed in the Nature paper in the brain. And with that, I'm going to hand over to my colleague, Filip.
Thank you. Do you see my screen now? Yeah, okay. So, hi everyone. Again, my name is Filip Sucharski. I'm preclinical scientist at Haleon, based in Lyon, and today I will present my understanding of the methodology used in the Nature paper by Nihart et al., which investigates microplastics and nanoplastics in human tissues. I will highlight key concerns and offer some scientific recommendations to improve future research based on my personal experience. Oops. Okay, so moving on to the methodology, we identified several areas of concern. Firstly, there is a risk of overestimation or underestimation due to residual biomatrix interference and lipid interference, particularly affecting the analysis of polyethylene. In pharma business, the bioanalytical studies, especially these which are going to be submitted to health authorities, require extensive method validation. And one of the important points of the method validation is matrix effect, which should be assessed, and the accepted levels of variability should be within plus minus 15% for all analytes. And going to the lecture that Cass presented, I think what you have done there was great, and that was for samples measured in the blood. But if we aim to analyze some other samples coming from different tissues, I think the method needs to be validated for every single metric, so meaning brain samples, etc. In pharma and those bioanalytical studies, we often use surrogate metrics. So, for instance, to analyze samples coming from brain tissues, like in the Nature paper, we would probably use brain tissues coming from the various animal species. Going to the next point, there is also a size and collection limitation. The use of ultracentrifugation may not only collect may not fully collect the nanoplastics, and the resolution of light microscopy is insufficient for visualizing nanoplastics smaller than 1 micron. So this is very strong limitation. Additionally, there was observed a significant analytical variability with 25% within sample variation across different laboratories. This should not really, this wouldn't be normally acceptable in the bioanalytical studies submitted to health authorities. Lastly, the study sample scope is limited because only one or single sample per organ was analyzed, and the geographical differences in MNP's concentrations were not fully accounted for. And so, going to the next slide, to improve the accuracy of MNP's quantification, we would recommend several scientific enhancements. First, employing advanced analytical techniques such as high-resolution mass spectrometry. That's the easiest solution always. This can provide better sensitivity and specificity. We could also propose to consider using internal standards labeled with stable isotopes, such as C13 or deuter, and these are widely used in bioanalytics in pharma industry. For sample preparation, and I think this is the most important part that should be investigated, some more rigorous biomatrix removal techniques can enhance nanoplastics isolation, such as extended enzymatic digestion using multiple enzymes, and also field flow fractionation technique seems to be a very interesting approach for this analytical challenge. Additionally, broadening the study design to include diverse samples from various geographical locations and analyzing multiple samples per organ will help assess distribution heterogeneity and improve the robustness of the findings. Thank you very much for your attention.
Thank you very much, Filip and Marianna. I now call on our next expert speaker, Dr. Inge Mulder. Please feel free to share your screen. A reminder, we will be getting back to the questions in the last part of the webinar.
All right, thank you. I guess you can see all my screen now.
Yes.
So I'm indeed Inge Mulder, assistant professor at the Amsterdam University Medical Center. And here in the lab, we're specialized in the vasculature of the brain. And I will give a brief insight in this vasculature and the micro and nanoplastics and their potential to breach the blood-brain barrier. So first of all, these are my disclosures. And as discussed in the previous talks, these MMPs, they have been found in the human brain, despite the fact that the brain is very well protected against these harmful substances by the blood-brain barrier. So how do these micro and nanoplastics actually enter the brain? So when we talk about the blood-brain barrier, I first have to explain a little bit how the vasculature of the brain is organized. So the large cerebral arteries, they are branching off from the circle of Willis, which is composed of at the basement of the brain, and then they branch into the large brain arteries, followed by the pyoarterioles, which are all located at the surface of the brain, and at one specific point they actually enter the brain, which is the penetrating arteriole. And then after that, they branch further into microvessels. And this is actually the point when we talk about the blood-brain barrier. So only in the capillaries of the brain. So this blood-brain barrier, it's actually composed, and it was touched upon briefly already by the previous speaker, but there are multiple physical barriers in this BBB. So when we go from the vascular lumen, when we start there, first, we have a layer of the glycocalyx, and this is a layer of glycosaminoglycans, and it has a netto negative charge, and this is actually functioning as a sort of a first barrier against ions or molecules with a negative charge. And then we get to the first cellular layer of the barrier, which is composed of endothelial cells, and they are very tightly connected to one another by these tight junctions and adherence junctions. Then there's a basement membrane and a second cellular layer, which is composed of the pericytes. And these pericytes are very important to regulate the transport, mainly among the endothelial cells. And they're also really closely connected to the endothelial cells by a PEG and socket connection. Then again, we have this basement membrane, and the last sort of physical barrier are the end feet of the astrocytes, and then we eventually end up in the brain tissue. So despite this very tight barrier, there is a lot of transport across this barrier, so into the brain tissue, but also clearing waste out of the brain tissue. And for this, there are multiple pathways for this selective transport. First of all, like in between the endothelial cells, so through the tight junctions, there are some selective transport of only water-soluble agents. As a second mechanism, there's this transcellular pathway, and only lipid-soluble agents can actually enter, because the membrane of these endothelial cells is composed of a lipid bilayer. So these lipid-soluble agents can fuse with the membrane and then go to the other side of this endothelial cell. The third option for transport is via transport proteins. And this is very selective for different molecules, for example, glucose. So they bind to these transport proteins and they are released into the cell and then travel through the other side. And then they are released again to go to the brain tissue. Then we have the receptor-mediated transcytose, for example, for insulin. And here the molecule binds at the luminal side of the endothelial cells, and they are internalized in these vesicles, and then are transported to the other side of the endothelial cells, where they are released again. And sort of the same mechanism is also there, but then for absorptive, it's called absorptive transcytosis, for example, for albumin, but at least it's positively charged molecules. So via these multiple pathways, compounds can still enter the brain. But the question is, can these MMPs also enter the brain via these pathways, or maybe do they use a different way? So in literature, there is not a lot of experimental evidence where they actually tested these MMPs in the central nervous system. So less than 30 articles, primary articles, showed some experimental evidence, and I tried to summarize them here for you all. So most papers talk about and conducted in vitro studies using mouse and human cell lines. Mainly these vascular endothelial cells were used, but also the effect of NMPs on astrocytes, microglia, and neurons was tested. And there were also a few papers, few in vivo papers, where they used mice, but also rats and zebrafish. And in these in vivo studies, the main administration route was orally, but also some intranasal experiments were done, or intravascular injections of MNPs. So a summary of what these articles showed as possible mechanisms. So it's again based on just limited evidence. But for the nanoplastics, what has been shown is that if these nanoplastics are in the bloodstream, there is the pointer. So nanoplastics in the bloodstream, they can disrupt the tight junctions, which results in leakage of the BBB, and these nanoplastics can actually enter the brain. The nanoplastics have also been shown to be able to be taken up by the cell membrane, so by the lipid bilayer of the cell membrane, whereafter they are internalized into the endothelial cells. And whether one or the other is cause or consequence, that's still unknown. But what was also shown in multiple papers, that the final effect of these microplastics is that there's a lot of oxidative stress in the endothelial cells. An inflammatory response is started, and there is a breakdown of the blood-brain barrier, and eventually this is causing neurological dysfunction and degeneration. And at the end, this is resulting in ischemia. So it's also very important, the different properties of the MNPs, which are of influence of this blood-brain barrier penetration. First of all, size is important. So the smaller, the faster they can actually penetrate into the brain. It was also shown that the polymer type is important. For example, PVC nanoparticles could be transported faster over the BBB compared to polystyrene nanoparticles. Shape was important, and also the inherent chemical composition and surface functionalization. So in one paper, they also showed that if there's a biological corona formed around these particles, it is of influence of, for example, the penetration into this lipid bilayer. So, for example, if you have a cholesterol corona, this actually facilitates the entry into the membrane layer, but it also traps these particles in the membrane of the cell and preventing to internalize it. So that was mainly on the nanoparticles, but what is happening with the microparticles, so relatively big particles when you talk about the blood-brain barrier and the biological cells composed of it. That's what we do in our lab, and here we use in vivo experiments. So we use mice with a cranial window, and by putting these mice under a 2-photon microscope, we can actually investigate the capillary network of these animals while the animal is still alive, and also track these individual microparticles. So this is an example. For example, the lumen, so the blood plasma, is colored by a fluorescent dye, which is the dextran, which is in green here, and then the microparticle is in red. And when we follow this microparticle over a few days, what we saw is that despite the quite large size of these microparticles, which is here 10 micrometer, they can still migrate through this capillary network, despite the fact that these capillaries have a smaller size. So these capillaries usually are from 5 to 8 micrometers in diameter. And this seems to be an active process that these particles can still migrate, and it's actually actively regulated by the endothelial cells. And what we see is that there is a temporary breach of the blood-brain barrier, so you see some leakage of the vascular dye, but this is also, again, restored after 1 or 2 days. So again, this same individual particle can travel for quite a long distance in this capillary network, despite the fact that at baseline, these capillaries and the diameters are quite a lot smaller than the diameter of the microparticle. And as a second effect, what we saw is even, I think, more interesting, is that extravasation. So at one point, these microparticles stop with their migration. And at the point where they stop, they are being pushed out of the vasculature by these endothelial cells. And you can see it a little bit better in 3D. And this is a process of 1 to a few days. So first, the microparticle is really trapped inside the capillary and totally closing off the blood flow. And after 1 or 2 days, it's already halfway. So part of the blood flow is restored again. And after a few days, we actually see that the whole particle has been pushed out of the vasculature and really is located in the brain tissue. And the important part of this process is that we don't see any leakage of this intravascular dye during this process. So that means that it's highly likely that this is a really tightly regulated process of the endothelial cells. And to look a little bit more into this process, we also do a lot of in vitro experiments. And this is an example of an in vitro endothelial cell culture with one of those microspheres. And here you can see that the endothelial cell here in red, so this is the membrane of the endothelial cell, is really enwrapping the microparticle and also the actin filaments, which is actually the skeleton of the endothelial cell. It's also forming and rearranging. And it's rearranging really nicely along the surface of these microparticles. And they're actually forming a pore. And this is the way we think that these microparticles can actually enter the brain. And this is a second example. Again, the actin in green. And you can really nicely see these 2 pores which are formed by the endothelial cell. And at actually the luminal side here on top, the endothelial cell is closing again. So they're really pushing out these particles to allow blood flow in the capillary again. So to add this to the possible mechanisms how these MNPs can actually enter the brain via the blood-brain barrier. So the microplastics, they can migrate through these smaller capillaries. But here, this temporal blood-brain barrier leakage is seen. And at a certain point, they can actually enter through a whole endothelial cells by the spore-forming process, but without the blood-brain barrier being broken down and without any leakage. And this means that at the end, it seems that there's still an intact vessel and an intact blood-brain barrier. Although the secondary effects when these bigger particles are in the brain tissue is still unknown. So what they do with the neurons or the astrocytes or the microglia. However, I didn't show it here, but what we also saw, if there are too many of these microparticles trapped in the capillary, blood flow is restricted above a certain threshold, and this induces hypoxia or hypoglycemia, which eventually can also end up with oxidative stress of the endothelial cells and the inflammatory response, and eventually neurological degeneration and ischemia. So I now mainly talked about the blood-brain barrier, because this is also the largest surface area of transport and molecular exchange from the blood to the brain. But it's also good to realize that there are many other barriers in the brain, how these MMPs can actually enter the brain. So for example, on the surface area, we have the arachnoid space with the glymphatic system and CSF, where the barrier is less tightly regulated. This is also in the ventricles, where the CSF is produced, and the blood-CSF barrier in the choroid plexus, which is also positioned in the ventricles. Here there are also barriers towards the brain, but they are less tightly regulated compared to the blood-brain barrier. And I won't go too much into detail. I also didn't find too much literature on this, but this is also an entry part where possible these micro and nanoparticles, for example, via the eye or via the nose, can eventually enter and end up into the brain. So yeah, that's what we do a lot in the lab. Mainly this was work of Kevin Moll, one of our PhD students, working a lot on these microplastics in the animal models. Thank you.
Thank you. Thank you very much, Dr. Mulder. I now call upon Dr. Dotquin. He's a proud member of and a scientific advisor of the Microplastics Initiative for ILSE Europe. I would like to give the mic to you, Todd, and please go ahead with the Q&A.
Perfect. See that there are a few questions that come through. I might want to take the opportunity to ask a couple of clarifying questions, maybe hopefully just short responses, but maybe for Inge, how did you introduce a 10 micron size particle into the mouse?
So together with the dye, we inject them intravenously or intraarterially, actually. So they are already in the bloodstream, and then we look where in the brain they end up and try to find them back with the microscope.
And you used presumably a fairly high dose.
Yeah.
Okay. And then for Cass, maybe just on the clarification on perhaps a couple of points. One is, I guess I understood that you do use the enzyme digestion method. So your matrix effects that you're evaluating are based on what happens after the digestion?
Yeah, so the end product of the digestion is basically a vial which contains blood, but also a lot of solvent and stuff like that, that we pour over the filter. So whatever gets stuck on the filter is contributing to the matrix effect. So yeah, what that exactly is of course, we don't know for sure, but yeah.
Yeah, because I think there's the point that Filip made was at least in that study, the brain study, that there was quite a lot. I think the relative contribution of the biological tissue in the brain was quite high. So they weren't able to remove, I think, a significant fraction of that, which again, perhaps introduces slightly different effect than what you were looking at in the blood. Because it is a bit surprising you didn't see a matrix effect with polyethylene, whereas I guess if you have such a high residue, I guess I'm just wondering if that might actually introduce a problem for polyethylene in that context.
Yeah, to comment a little bit on the brain work, they used not enzymatic digestion, but digestion with nitric acid. And that sample was centrifuged, and then the pilot was taken out, and that was analyzed as a whole. So that is, at one point, it has an advantage because you're not limited by the pore size of your filter, right? You can measure nanoplastics all the way down to how small you they can be. But of course, what is really remaining in this palette is a bit unclear.
Yeah. Also interesting to know in the brain study was that the residue that was in the brain was higher than what was in the kidney and liver, because they also report data for the and the relative difference, it seems, in the concentration of polyethylene is somewhat consistent with the difference in.
Okay. Yeah.
And one final point I might clarify on the brain study, which Filip didn't mention, was that the tissues were dissected on polyethylene cutting boards, and that's clearly defined in the supplementary information of that paper. And the only polymer that they found was polyethylene. So in addition to the potential matrix effects, that there is also this additional sort of challenge, I think, that's associated with that study. Along those lines, maybe I'll sort of ask a question that comes from Rob Ellis Hutchings, sort of along these lines, which is perhaps related to best practices. I don't know, that's Cass, Frederic, if you can provide any guidance or thoughts about what might represent best practice for using pyrolysis GCM methods, particularly in the publication space. So I guess in terms of editors or peer reviewers, it would be helpful perhaps to have a checklist of things that people could be checking for.
Maybe I can start. Cass, please feel free to complement. Well, I also saw later on. There's also a question related to the feasibility or the accuracy of pyrolysis GCMS with respect to spectroscopic techniques. I mean, from an ideal perspective, I think that both spectroscopic and spectrometric techniques should be applied in parallel. The issue, obviously, of the spectroscopic one is the lack of high throughput, while pyrolysis allows you to process larger number of samples in a shorter amount of time. And I think when it comes to the Q&A and QA and QC of pyrolysis in particular, I think that a thorough method validation is really essential. And what Cass has been showing with all the experiments that we did in our work with evaluating matrix effect, evaluating these effects specifically on the blood matrix. And that's also something that Filip was mentioning afterwards, the importance of doing this for various matrices. This is also essential. It has to be done for each type of matrix and also for the different types of polymers that are being analyzed. And at the time being, there is no guideline, but you, Todd, are aware as well. There is some initiatives on that side to come up with a certain number of criteria that need to be fulfilled in order to provide a transparent overview of how the results were generated. And that goes from a thorough analysis of blanks, having control samples, providing information about the matrix effects, and so forth. So these are things that are needed, and we are working on it. And I think that the work that Cass presented is a pretty good starting. There's still a lot that we need to do, but it's definitely a good starting point towards achieving that.
Yeah, and I see we're kind of almost up against the time. I almost feel like that comment perhaps also reflects the question around the oligomers, and I guess having good methods will hopefully help to address the potential influence or the interference that might be associated with the oligomers. I just maybe want to end with it's more a thought around, we've been exposed, I guess, as a species to particulates, particularly nano-sized black carbon, for instance, for a long, long time. Do we find evidence that these particles bioaccumulate in the brain, for instance? Are they circulating the blood? Because I know there's a strong emphasis on the nano microplastic particles, but are there any other particles that are capable of this functionality, or is there something novel about micro nanoplastic? It might be for the brain more, the people studying the blood-brain barrier, but from a pharmaceutical perspective, pharma has been spending a lot of money to try to use nanotechnology to deliver a drug, an active ingredient, and yet struggling with that, and yet microplastic seem to have a capability of doing this. Have they missed something?
If I can respond to this, I think when you want to deliver drugs, you want to have it in a certain amount and also quite fast. And when we talk about these environmental micro and nanoplastics, it's more of a chronic thing where that they are in the blood. And what I showed and what we do in the lab here as well, we are talking about also days that they can actually enter the brain. So and we're still in the like doing the first steps with these experiments, but I think when you talk about drug delivery, the process might be, for what we now see, a bit too slow to be really effective when you talk about extravasation. And then for drug delivery, it might be better to choose different pathways to get the drugs in the brain tissue.
Or what about other naturally? So if I'm going to have a barbecue tonight, I'm sure it's going to have a lot of black carbon in it. We have a chronic exposure to other types of particulates in this.
I actually don't know about these other types or particles and any evidence that they have been found in the brain.
Okay. I think that's put us just over time, unfortunately, but I think I saw a message. If your question wasn't answered, hopefully, yeah, there's a response to email. Get in touch with the speaker. Yeah.
Yes, indeed. Well, to wrap up, thank you very much. Thank you for your time, for everybody, and especially the speakers today, for your efforts and collaborating and aligning so amazingly on this webinar. We look forward to collaborate with you again and to be a part of these webinars. Please get in touch to me or the Community of Practice of Europe, initiated by Microplastics Initiative. Thank you very much for your time today, and I hope you have a great day ahead. Thank you.
Thank you all. Thank you
Thank you
for
very much.
hosting.
Thanks a lot.
Thank you.
Bye.
Bye-bye.
Thank you, Todd. Thank you very much.
Bye.
Bye-bye.